Hello, everyone. I wanted to talk about this email. It was one of the very first emails when I originally contacted Patrick Jordan almost 3 years ago, and um, it's about six-letter DNA. Six-letter DNA alphabet produces proteins in cells from the Scientist magazine. So I'm going to read this article, and then we'll look at what Patrick had to say back. Uh, well, hold on. Okay. A synthetic base pair, first reported in 2014, uh, can now not only replicate inside living cells, but also encode and produce proteins containing atypical amino acids, according to a report in Nature today, November 29. This proof of principle advance now sets the stage for biochemists to generate proteins with entirely novel forms and functions to those that can be created by natural organisms, say the authors. What a beautiful paper, says chemical and biological engineer Michael Jewett of Northwestern University, who was not involved in the study. What's so special about the work is that the authors have captured the entire information flow of the central dogma, information storage, retrieval, and ultimately translation into a functional output using this expanded genetic alphabet. In all life forms on Earth, genetic information is composed of a four-letter alphabet, the nucleotides G, C, A, and T, which form the base pairs G, C, and AT. But 3 years ago, chemistry professor Floyd Romesberg of the Scripps Research Institute in California and colleagues extended this alphabet. Interesting. Uh, Scripts, like as in rewrite the script. Uh, Reporting the creation of additional artificial nucleotides X and Y that could pair up with DNA and take part in replication within a living bacterial cell. All right, did you get that? That this chemistry professor, Floyd Romesberg of the Scripps Research Institute in California and colleagues, extended this alphabet, the alphabet of the four letters that the creator uh, created. They have now inserted two additional artificial nucleotides, X and Y, that could pair up within the DNA and take part in replication within a living bacterial cell. From that point on, Romesberg explains, the team's ambition was to get the molecules to function within polymerases and with ribosomes in a cell. That is, work with the cellular machinery that transcribes DNA into RNA and translates RNA into protein. Now, that ambition has been achieved. First, the researchers introduced the artificial base pair X and Y into the gene for green fluorescent protein, switching a codon in a non-critical part of the gene from TAC, which encodes the amino acid tyrosine, to AXC. Next, they created a transfer RNA that contained the corresponding anticodon, GYT, that carried a non-canonical amino acid, canonical. Where have we heard that word before? Canon, as in scripture. So non-canonical would be not conforming to the canon, which is our original four base. Anyways, I digress. That carried a non-canonical amino acid called PRK, a research supplied amino acid that is rarely found in any natural proteins. The team then expressed these two genes inside specialized bacteria that support the retention of synthetic nucleotides within their DNA, called semi-synthetic organisms. Lo and behold, the microbes produce green fluorescent protein containing the non-standard amino acid. It's an engineering feat, says biologist and biochemist Eugene Wu of the University of Richmond in Virginia, who did not participate in the research. It is a surprise that everything worked so well, adds biological chemist Nigel Richards of Cardiff University in the UK. It's such a complicated system, and there's so many ways where it could have failed, but it didn't. The team went on to show that the transcription and translation could occur within an alternate synthetic codon, GXC, and result in the inclusion of yet another non canonical amino acid called PAZF. They used several assays, including mass spectrometry, I'm sorry, that tongue ties me, and click chemistry to confirm the presence of the non-canonical amino acids within the proteins. The artificial XY base pair is formed via hydrophobic attraction between the two elements rather than hydrogen bonding, which normally forms the connections between the natural Watson and Crick base pairs. But the X and Y nucleotides are otherwise similar, sharing the sugar phosphate base composition of normal nucleotides. It's really interesting that you don't need hydrogen bonding to control information transfer, says Richards. What it tells me, says Wu, is that being able to approximate the shape of base pairs is enough. However, the unusual pairing chemistry likely limits the number of artificial base pairs that could be included in a DNA molecule, says Richards. You get distortions in the helix where the base pairs occur, he explained. So a single base pair may be accommodated because the surrounding Watson and Crick base pairs almost certainly compensate. But if you had three in a row, then now it's not so clear that you could maintain the helical structure or that the enzymes would actually work. Other researchers, including Steve Benner of the Foundation for Applied Molecular Evolution in Florida, have created a number of novel base pairs that do utilize hydrogen bonding. These integrate into DNA without disrupting the double helix and can be present within the DNA in long stretches. However, so far, these nucleotides are only capable of replication, transcription, and translation in vitro, Brenner explains in an email to the scientist. Most likely application for Romesberg's artificial base pair is to incorporate unnatural amino acids into specific parts of proteins. Unnatural, where the natural becomes unnatural. Incorporate unnatural amino acids into specific parts of protein, says Wu, thereby vastly increasing the possibilities for biochemists to create proteins with new functions. Having a unique amino acid within a protein could, for example, enable the attachment of a drug or other molecule of interest to a specific point on a specific protein. And for those sorts of applications, the limitations of hydrophobic bonding probably doesn't matter, said Richards. Ultimately, the XY engineers wanted to get molecules that function in a cell. That was our focus, says Romesberg. Before this paper, he adds, every protein produced in any living cell has been produced by decoding a 4-letter alphabet. We now have reported that decoding of proteins with a 6-letter alphabet that makes the hair stand up on the back of my neck, he says. Yeah, it makes the hair stand up on the back of my neck as well, but likely not for the very same reasons. So here I will put the link. Actually, I have the link into the entire article, the semi-synthetic organism that stores and retrieves increased genetic information from the Nature article I will put down below. So let's see Patrick's response to he says, I find myself repeating myself way too often. I did, however, get counseling from a red hen that since people are lost in religious delusion, that when I try to span the gap between mythos and science, that they, in their inability to grasp reality, think that I'm merely promoting my own made-up religion, so they try to bring me to Jeebus. Consider again with me that Ebola is just a demon genetic code that attempt to possess the host, but is incompatible with this dimension. So it kills the host. Thing of it is, despite the hype of Ebola and Marburg, they don't kill everyone all the time. That is what it was thought. Tolerance is not a good thing. Blessed are the babies that die upon vaccination, because that means that they were too pure to accept a demonic possession. Seems harsh, I know, but it's my religion of mechanism, and it isn't a joke. So it is simple to create artificial base pairs. Of course, it would make novel proteins that already had a name 30 years ago, never-born proteins, thus sealing the circle of the Ouroboros speaking itself into existence. So we go full circle of the Archon virus being a biological entity that drives the host to do its bidding to create a synthetic biological entities that will allow it purchase on this reality that will culminate in the artificial intelligence being a metamorphosis final stage of development that will be as impossible as a sporulated encysted parasite to be destroyed once it has completed that cycle. All with willing, eager college puke fucks doing the work to please their masters for a scribbled letter grade on a piece of paper. God save the world from my wrath if I could steer comets. And they used to burn witches in the public square based on the whisper of cavorting with the devil. So back in May, when all this started, I brought this to his attention again, because I started looking at this and the Scripps research, but then I read on Romesberg and his colleagues. Let's see. Well, let's just read this little part. Okay. This is from Augmenting the Genetic Alphabet. Living organisms have two sets of DNA base pairs, adenine paired with thymine and cytosine paired with guanine, that together encode the 20 amino acids used to make proteins in the cell. Now, scientists at the Scripps Research Hoyle, California, have introduced a synthetic base pair to E. coli, greatly expanding the information DNA is able to encode. DNA containing the novel base pair can replicate within the bacteria, according to a paper published today, May 7th, in Nature. I commented Scripps, interesting, and as in rewriting the script. Romesberg and his colleagues are now working to get RNA polymerases to transcribe the novel DNA into messenger RNAs. Then they hope to engineer cells to transcribe the transfer RNA that will read novel codons and translate novel amino acid sequences into proteins. Romesberg suggested that synthetic biologists will eventually build proteins with both natural amino acids currently not used in proteins and amino acids synthesized in the lab. He added that Synthor, a company created based on his work and which he was scientific founder, aims to ramp up the production of synthetic triphosphates. So when I noticed Synthor, and I can't help but see Sin, Thor, and then X, I looked them up and I found out that they were literally just purchased by Sanofi. Let's see what is there a date on this? Okay, I think this is Francis Sanofi on Monday agreed to buy California headquartered biotechnology firm Synthor in a cash deal worth $2.5 billion. Sanofi is stepping up a push in the lucrative field of cancer drugs under its new chief executive, and Sanofi offered to buy all the outstanding shares of Synthor common stock for 68 cents per share in cash, 172% premium to Synthor's closing price on December 2019. So let's see, did I get... I was trying to see if I had already opened something, but I can't find. Yeah, here we go. Mega deal. Synthor sells for $2.5 billion. So now what I did then, let's see if it's here. So I typed in Sanofi and what do they have to do with COVID-19? And obviously, of course they do. Finance here, Yahoo Finance they just launched trial for COVID-19 protein-based vaccine. So let's see. We'll go here to this Stat News and see what they have to say about these vaccine human trials and what is Sanofi and GSK's COVID-19 vaccine. And I wonder, is Sanofi using their new nucleotides X and Y in this, how that plays a part in this. Vaccine giants Sanofi and GSK are joining the ranks of COVID-19 vaccine makers, blah, blah, blah. Phase 1 and 2, first in human trials. What is it? It's combined with an adjuvant, a compound that enhances the immune response to the vaccine produced by GSK. Like most COVID-19 vaccines. Okay, the 3 candidates already in phase 3 trials in the U.S. use vaccine platforms that have not produced licensed vaccines. Moderna and Pfizer use a messenger RNA approach, while a vaccine being developed at Oxford University and AstraZeneca use a harmless adenovirus into which genetic material from the SARS-CoV-2 virus, which causes COVID-19, has been fused. The adenovirus, which doesn't trigger illness, induces the immune system to generate protective response. Unlike the mRNA vaccines, which need to be transported and stored frozen, the Sanofi vaccine will be shipped in liquid form and can be stored at refrigerator temperatures between 2 Celsius and 8 Celsius, Shiver said. The Pfizer vaccine must be stored at negative 70 degrees Celsius, which will complicate delivery. Sanofi is also working on a second vaccine, an mRNA vaccine, which is making in partnership with Translate Bio Human. Translate Bio, as a transformation, transfect. In fact, let's look at this. Patrick sent me this link. I actually read this in his book, and unfortunately, I don't have a link to it, but I'll read you Patrick's Transfection. And again, this is from his book, Prion Agenda. Transcription. Instructions from DNA are copied by transfer RNA that's then shuttled to the endoplasmic reticulum to build proteins. Transcription is the translation by transfer RNA. Whoever the red-headed stepchild is that developed this naming structure needs to be beaten in public. Transduce. The transfer of genetic material from one bacterial cell to another bacterial cell, a process often accomplished by a bacteriophage that injects chunks of bacterial chromosomes but no viral DNA, or by a plasmid, which is circular, highly infectious, transducible DNA. Transfect, as per my schizophrenic dictionary, is to infect a cell with bacteriophage, animal, or plant virus so that the infected cell replicates into a complete virus. But the multiple industrial sources that do this shit for money indicate that it's primary a non-viral method of getting nucleic material into a cell using things like plasmids, lipids, liposomal anything ought to make your testicle crawl, reagent-created nanoparticles, to chaperone plasmids, microinjection, and electroporation. Still want to use Bob Beck's electroporation pulsar. Since these folks take your money, they don't care about the dictionary definition. If you want your viral RNA stuck up the ass of a cell, then by God, that's what you'll get. Lay your money down. Transient transfection is a process that gets plasmids and expressible genes into a cell without incorporating it into the host chromosome. Polyethylenamine, PEI, and calcium phosphate are used as vehicles to get this shit inside of cells. I would read doof labels from now on to make sure you aren't facilitating your own rewrites by eating prepared foods. Stable transfection gets the nasty bits installed in the cell as an episome or inserted into the host chromosomes, like putting a cat inside a mailbox. That used to be a prank out in the country of the grain ghetto. Believe me, you don't want to be the one opening that box. In order to make sure that the cat got into the right address, they used markers, one of which was being dihydrofolate reductase. That alone should make your whiskers tweak, but if it doesn't, then DHFR might get you closer to the MTHFR. I always see motherfucker when I see that. So when you see that what they're doing in the lab is indistinguishable from what they're doing on the street. Oddly, a corporation specializing in transfection procedures says that stable transfection is rare. transform, to genetically alter a cell with free, naked, foreign DNA from another cell or virus. This is the essence of what everyone fears from GMOs, to put the gene for human insulin into bacteria so that it overproduces that industrial product for harvest. Converting normal cells into immortal lines of cancer cells is called transformation. The HeLa cells from Henrietta Lacks cervical cancer can infect, transfect, transform whatever they come in contact uh, with to become the undead. Transformation: when an oncoretrovirus inserts itself into the host genome, it converts that cell into a cancer. A cancer is a new species. It's a parasite that replicates uncontrollably at the expense of the host. When a cell is transformed, it's infected, converted, and cancerous. It's then said to be immortal. It will continue to grow and reproduce as long as there is food supply. The nutrient requirements for normal human cells is complex. Cancer cells have minimal food needs. Translation: DNA is a code. RNA is a mirror of that code that is read at the endoplasmic reticulum that translates the code into a series of amino acids that are strung together to make proteins. This is literally the language of our life, of our bodies. Translocation: musical chair of genes. This can occur in the host genome or under the influence of an infectious agent. Transvect, like vector, venom, vector, coined in the genetic sense by Edward B. Lewis in 1954, an epigenetic interaction between an allele on one chromosome and the corresponding allele on the homologous chromosome. Transfection effects include both activation and silencing. The act of supernatural flight, such as By Which, 1996, Abner Falk, A Psychoanalytic History of Jews, ISBN 083 863 6608, page 532, in Transfection, Witches Rode Broomsticks, a classical phallic symbol. Definitely witchcraft going on here. Transfection: insertion of genetic material by a bacteriophage virus. This can be the phage inserting its own material into the bacteria to take over that cell's function, or it can be the artificial manipulation of a phage to carry genetic material that was put into it by the military. Transfection was the process that revolutionized biology and biophysics to become molecular biology. It was the beginning of genetic modification. The classic T phages can be thought of as syringes that insert genes into a host using restriction enzymes to insert those gene sequences into the host nucleus, and then using the host machinery to make copies of itself. Once this process was understood, then all of life was reduced to an engineering problem that could be solved by science. tRNA, or transfer RNA, messenger RNA. Messenger is the copy of DNA that gets taken to the endoplasmic reticulum to be decoded, translated, a confusing damn words starting with T, into functional molecules with transfer RNA, shuttling amino acids to the RNA in the ribosome factory for assembly. Wow. Yes. Another popular option is the use of transfection reagent. Several companies offer specialized short interfering RNA delivery reagents. Please consult the transfection reagent vendor's protocol for detailed information for the exact requirements and procedure. Careful optimization of variable factors should be ensured for all initial transfection experiment. It's based on this and further optimization that reproducible gene knockout results will be obtained. Usually, RAIN, or RNA interference effect, is seen within 4 hours, and the maximum knockdown regulation observed 24 to 48 hours. So I just wanted to remind you, since we were talking about these words, transfection, transformation, and I'm just trying to give you more evidence of the literal rewrite, the reprogramming, the rewriting of humanity. This is absolutely no joke. Let me know what your thoughts are. See you soon.